The overall goal of this protocol is to use PathWhiz and the Internet to easily create, replicate, and propagate biochemical pathways for a variety of purposes and applications.
This tool allows scientists, educators, and students to create visually pleasing and accurate biochemical pathways with a high level of biological detail and complexity. PathWhiz is a free public gallery with thousands of pathways and pathway components, which is compatible with essentially any operating system and can be accessed and expanded upon by all users.
Begin by loading the PathWhiz page in any modern web browser and clicking on the Draw menu. Click the New Pathway button to start a new pathway and enter the name of the pathway to be drawn. Choose the type of pathway from the drop-down list. Then enter the scientific name of the organism into the autocomplete box to search for the organism of interest, followed by the scientific name of the species and the common name. Select the species classification from the drop-down list as eukaryote or prokaryote, and enter the taxonomy ID from the NCBI taxonomy. Click Create Species and enter a comprehensive description for the pathway, clicking Add Reference to add external references to the pathway as appropriate. Next, click Create Pathway. A white gridded canvas will appear with a gray menu bar. Click the Add Process link and select Add Reaction to add the first process visualization. Enter the reactants, products, or enzymes of interest in the autocomplete box to search the existing reactions.